Good night and welcome everyone to our discussion on viral load analysis of honeybees in the Caribbean. My name is Dr. Sharda Mahabir and I will act as moderator for tonight's session. And to begin our session tonight, I will actually give you a little bit of background as to how this project came about. And in order to do that, I have a presentation entitled South-South Regional Apiculture and Biodiversity Project. So in 2020, JFSGP received funding to complete a South-South cooperative project between countries in the Caribbean and the Pacific. The participating countries at the beginning of the project were actually only 7: Dominica, Grenada, St. Kitts and Nevis, St. Lucia, St. Vincent, Trinidad and Tobago, and Samoa. However, as we progressed with the project, we were able to also collect data from Antigua and Barbuda in particular, which was actually added to our project report, and also collect some baseline data for Anguilla as well. So because of this cooperation and the interaction of beekeepers on these islands, we started to ask some important questions, which we will look at. So for this project, the JFSGP partnered with Iyanola Apiculture Collective to complete a baseline study of beekeepers across the Caribbean. This baseline study covered two important elements: the study of the practices, so the knowledge, the attitudes, the performances of beekeepers. So how do they carry out their beekeeping in terms of different methods, techniques, and specifically what diseases they see on their islands and how do they treat with them. In terms of the complete survey, that it consisted of 20 questions that were provided to all the beekeepers across the Caribbean, and the questions were actually in different categories. The first category, of course, is baseline demographic information about the beekeepers, their age, experiences, etc. The type of apiaries that they have. Do they have how many hives do they have, and are they located in agricultural spaces or in forested spaces, etc. Their knowledge of apiculture, so some information about honeybee biology. Can they identify the different castes of bees? Can they identify diseases of bees? And of course, the last category of questions we asked was in terms of honey production. Generally, what kind of production were they having in terms of liters per season or kilograms per season, as well as if they actually dabbled or developed any value-added products. Now, in the baseline study for the entire of the Caribbean, for all 8 countries, we had a total of 193 responses, which technically represented about 25% of the beekeepers for the entire Caribbean, which is a very good representation. Overall, we had some exceptional responses from St. Kitts, but overall, we had somewhere between 20 and 30% averaging for most of the other countries that were represented, except of course for Grenada. That was also of a significant, that 50% of their population responding. Now, with respect to the viral load testing that we are going to talk about today, in each country, the beekeeping organizations were contacted to participate in the overall survey activities, which is including the baseline survey, the questionnaire that I just shared with you. In Trinidad and Tobago, JFSGP hosted a meeting with Trinidad and Tobago Beekeeping Association and the Tobago Apiculture Society to drum up support for the survey activities, and persons were encouraged to contact us if they wanted to participate in the survey. Unfortunately, only 3 persons contacted JEF, and their locations were used in the preliminary analysis of the viral load testing for Trinidad and Tobago. Now, these 3 persons represent 7 apiaries across the entire island, which we will see shortly. So this is a map of the location of the apiaries, general location of the apiaries for the participants in the survey. As you can see, it's a widespread covering both North and South Trinidad. It was widely distributed across the island. So with that, I'd like to thank you very much for allowing me to give you a quick synopsis of how this all started. And without any further ado, I would like to introduce you to Dr. David Tarpy. He is a professor and extension apiculturalist within the Department of Applied Ecology at the University of North Carolina State University in North Carolina. Dr. Tarpy came recommended from Cornell University for his extensive work with respect to honeybee testing, and because his lab is accredited, and he was contacted for this project in order to be able to give us accurate data on viral load found in honeybee samples that we collected in the 7 apiaries in those 4 locations. So I would like to extend thank you to Dr. Tarpy for taking the time tonight.
That varroa mites don't actually kill your bees. It's the viruses that they vector that kill the bees. And there's lots of different honeybee viruses, and they've always been there in the system, in our honeybees, but to a greater or lesser degree. And the varroa is actually what's making them a lot more pathogenic. So this is a non-comprehensive list of the different viruses that have been identified in honeybees. And it just shows that there's lots of them there, and they are they're vectored, they're passed from one individual to another in many different ways, and they can be passed to each other by feeding and by interacting with each other, or they can be passed from the mother queen to the subsequent daughters through the egg as she's laying the egg. Now you introduce varroa into this, the mix, they act like hypodermic syringe needles, right? Where they're kind of transferring these viruses from one individual to another. And so they kind of heighten the sharing of these different pathogens within colonies. And so that's what really makes them very devastating. So the calling card of varroa mite, the thing that a lot of people identify with varroa parasitism, is the deformed wings, the crinkled wings like this, where the bees emerge and they can't fly, that they're, because of varroa parasitism. And while that's true, the deformed wings is not actually from varroa itself. It's from a virus that they're vectoring, a virus that they're transmitting to the bees. And so this is almost ubiquitous, at least here in the United States. There isn't a colony practically that you can't find, that doesn't have some degree of deformed wing virus in it. But there are others. So this is another notable one, black queen cell virus, where a lot of colonies have this, a lot of workers within those colonies have this particular virus. It really is only symptomatic when you're trying to raise queens. And so within the queen cell, the larva dies, or the pupa dies, and then it kind of melts and becomes molten and turns blackish and decaying. But it is very highly prevalent in a lot of colonies, at least in the continental US. And there was a study done in Canada where it was the #1 marker and predictor of winter mortality. And so it really does have a profound effect on the colony, even though there's not really any symptoms among the workers, which makes it very, very insidious. This is another virus. It's called chronic bee paralysis virus. If you've ever seen bees that look kind of oily and darkened, especially in their abdomens, like this one right here, that's probably from this particular virus. It can even sometimes mimic pesticide poisonings, where the bees can't fly and they shake and are kind of agitated, and they just look very sickly. This is relatively more rare among the viruses, but it can be very, very problematic if a colony gets it at high levels. And then there's these three viruses that are very closely related to each other. They're very, they're kind of kissing cousins to each other. The acute bee paralysis virus, the Kashmir bee virus, and the Israeli acute paralysis virus. They're all very similar to each other, kind of genetically related. They all not really sure what their symptoms have or what their symptoms are, but they are associated with increased colony mortality and death. And in some cases, their prevalence in colonies is fairly spotty. Now, that's just a very quick overview of the viruses, because I want to make sure that we can cover the other speakers and have more time for discussion. But pretty much everywhere where Varroa has been introduced, they facilitate the spread of these viruses that are already there, and they facilitate the selection for more virulent strains. So deformed wing virus, as I said before, is kind of the calling card of Varroa. Well, different studies have shown that when Varroa is introduced to a system, the relatively less virulent form of that virus, deformed wing A, is replaced by the more virulent form of it, deformed wing strain B. And so, the more that these things occur and are active within a given population, you can see these once relatively benign viruses getting worse and worse and worse. Now, when you have the accumulation of all of these different viruses, when you have lots of mites in the system, the colony can often result in the population collapses and crashes, and the colony can often die from this parasitic mite syndrome. So again, it's just too many mites passing along too many viruses, and it just is strongly associated, one of the leading causes of winter mortality up here in the continental US. But you can also have that same problem, but just without high levels of Varroa. And this is called idiopathic brood disease syndrome, which is in essence, the viruses are there, but the mites are not. So you can still have this kind of accumulation of too many pathogens within the colony, and the colony simply collapses, even if the Varroa mites are not at high levels or not present or abundant. So again, it makes it very complicated to be able to tease all of these different things apart, especially when these viruses are invisible or asymptomatic. They're there and you just don't know it. And so really the only way to be able to determine that is using some of these genetic techniques. So that's why we developed this queen and disease clinic. So we have two branches of this. The queen side is where we measure queens for their sperm viability and some other health measures. But then the disease side is what Richard had reached out to me about, to see if we could screen some colonies in Trinidad and Tobago to see what types of pathogens they have, because there isn't really any published report or other data to that effect. And so again, it took a little bit of doing, quite a lot, as Sharda had explained, to get those samples, but we were able to receive them and run them. And so I'd like to share them with you here right now. In fact, let me sorry, I wanted to. Okay. All right, that's better. All right. So this is the report that we had provided Richard. And let me just walk you through this, because this is kind of the standard report that we provide anybody who sends us samples. So this graphic up here on the top right is a baseline sample of all of the different colonies that we have measured that have been sent to the clinic, and that's almost 1,700 samples. So it's one of the largest baseline sample sizes that we have in the United States. And then these are the different targets, the different pathogens down here, which again are these different viruses, many of which I went over, especially these deformed wing virus version A and strain B. And so the percentages, the blank part, the white part, is where there's very little detection of that particular virus. But then the light gray, medium gray, and dark black represent the low, medium, and high levels of these different viruses. So if we take deformed wing A, for example, about 35% of them were infected with some level of deformed wing virus. But this is how many had low levels of detection. This is how many had medium levels of detection. And this is how many of them had extremely high levels of detection. And then, so that's true for all of these different pathogens. Now, again, while any one single pathogen might be, only about 35 or so detected in about 33% of the colonies, pretty much every single colony has at least one, if not multiple, types of these different viruses that are screened here. So, so anyway, that represents the baseline. That's kind of the expected of what we tend to see in samples that are sent to us. Down below here are the results from the submitted sample. In this case, there were 8 colonies, if I believe. And it just goes over those same targets and what we found in those samples, comparing them to the baseline. So for acute bee paralysis, most were not detected, but we did detect one that had relatively medium levels of that virus. For black queen cell virus, we found one colony that had high levels, one colony that had medium levels, but otherwise not detected. There were none for chronic bee paralysis virus that were detected. The 2 deformed wing viruses, we had the most detections, as we might expect in other populations, where there were several that had medium or low levels of deformed wing A, and there were several that had extremely high levels of this more virulent form, deformed wing B. And so the Israeli acute paralysis virus, again, some levels, but relatively low. No Lake Sinai viruses, and these other 2 pathogens that I didn't cover for the sake of time that are also present, but not necessarily in higher levels. So when we go to the second page of that report, it's broken down by each individual colony, and then you can see what those levels are, again, using that same color scheme to show what is found at these different regions. Now, again, Sharda had briefly mentioned where some of these were taken, and you might be able to correlate that with management practices and location and other things, but with such a small sample size, probably can't make very strong inferences about that. But I'll just make a couple general comments here. First and foremost, the levels of nosema, which is a gut parasite, not a virus, but it's something that lives in the gut, was ubiquitous among your colonies in Trinidad and Tobago, which is actually fairly rare. It's actually higher than what we might expect based on the baseline data here in the United States. Another inference is that your colonies in the Caribbean are not unlike our colonies here in the continental US or elsewhere in the world, in that they have these viruses and they can have them at fairly high levels. So, the pathogen ecology is actually fairly similar, although we don't know if it's the same, and we don't know, what the different dynamics are. The third take-home message is that it does seem like this deformed wing B virus might be more virulent than the deformed wing A, which is indicative that the varroa mite has indeed been selecting for these more virulent strains. So it really kind of goes to show that keeping and managing the varroa mite is very critical. So as yet, just having 8 colonies is just a drop in the bucket. There are many, many, many unknowns. And so why we're having this discussion, because we need, I would think, a better picture to see what are the different pathogens in the different countries and islands in the Caribbean, and are there any differences in those different populations, and what are their effects? What are the dynamics with varroa mites and management practices and other things that you do locally? And what are some ways to perhaps mitigate these different pathogens? You also have Africanized honeybees in many of the islands, and some do and some don't. So what is the difference between there? We really have no idea, and that would be interesting to know. So, I'm looking at this is a real opportunity to try to secure some grant funding or other support to be able to do a much larger scale systematic sampling of the different nations and islands in the Caribbean, to be able to try to make some more stronger inferences about how managing and mitigating these different pathogens might help improve colony survival and overall colony productivity. And so with that, I'd be happy to turn that over. I think I made it back on time. We're at 7:35, so that the other speakers can have their turn.
Thank you so much, David, and for that very concise and very thorough a summary of the results that we've seen. I just want to take a minute to make sure that, we have had so many people come in during the session that I wanted to be sure that I capture some of the succinct points, so correct me if I'm wrong. So one of the first things that you indicated was that varroa was a main transmitter or vector for many of the viruses that you saw in your samples. That's the first thing. The second thing is that the viruses themselves, the varroa itself will not kill the bees or the colony, but the combination of the viruses as well as the varroa weakens the bees enough to potentially, weaken the colony to the point where it may not be able to survive. That's the second most important thing. The third thing is that you went through about 4 different diseases: deformed wing virus, parasitic something virus, black queen virus, etc., all of which were found in Trinidad and Tobago. It's about 6 of them in total, if I'm not mistaken. And what you're saying is that those viruses are directly related to varroa, which is your first point. Okay. Yes. So in other words, varroa is not just a pest. It's not like a tick alone. It is actually bringing into or introducing into the honeybee viruses. But the fourth and most important point is that it may actually be helping these viruses to get worse, to become more virulent. So it's preferring the more aggressive strains of these viruses, right? I think one of the other things that you highlighted was that the management of your bees is important in terms of managing those diseases as well as varroa. So varroa is directly not related to, but is sort of linked to these diseases, but these diseases could actually be in the bees without actually the varroa being there.
Correct. Right. And the varroa just makes them worse.
Right. So I hope our attentive audience is taking notes as well. because what we're basically saying this evening is that, you know, traditionally in Trinidad and Tobago, we thought that the only real disease that affected bees was varroa. That's it. Now we have at least 6 or 7 more diseases that you've shared with us that you're saying are now present in Trinidad and also present at different locations of the country. The most important thing that I think you said, and we didn't really discuss it in detail, was the nosema. Now, nosema in itself, from what I understand, is a very potent condition. I don't want to say disease, but condition of bees, because it in itself can cause a lot of damage as well. And
Correct.
of course,
Yes.
nosema was another main disease that was found across the entire island. So what we're basically saying is that our bees in Trinidad and Tobago may be a threat to these new viruses that may be more prevalent than we think. Oh, and the 6th most important point is that the symptoms of these diseases are not visible. The bee doesn't turn green, you know, it doesn't, its eyes doesn't turn purple, you know, there's no fluorescence, nothing like that to say that we know that our bees have these viruses. So the thing is, these diseases are moving in individually, you know, like a ghost in the night through our biodiversity, especially in our honeybees, and they're affecting them. One quick question. You know, Trinidad and Tobago has not really been able to meet its local production for honey. Can you say that the presence of these diseases, especially the fact that we have a lot of new viral diseases we're recognizing through this testing, as well as the nosema, which I believe is a fungi.
It's a gut fungus, yes.
Right. So now that we've seen all of these diseases, could this actually be impacting the ability of the island to produce enough honey for itself?
Absolutely. So let's just take deformed wing virus as a good example, right? The bee emerges without wings. How good a nectar forager do you think she's going to be, right? So as these levels build up within the colony, they're clearly not going to be making the honey, you know, the surplus honey that they would otherwise. So, you know, these pathogen, nosema is the same way. They can just get so laden with these gut parasites that they're literally too heavy to fly. And so again, that has a direct impact on their productivity and on the honey yield. So absolutely, healthy bees result in productive bees.
Okay, and just one last question. I mean, in your experience or expertise, what advice would you say would be best in terms of being able to address all of these diseases that we've identified? I mean, in terms of, you know, how best can we manage these diseases?
So I really have 3 answers to that, and I'll try to make them quick. The first is knowing what you have. So that's part of the understanding, is I think having a more systematic survey that's more comprehensive and of greater coverage, both geographically and temporally, to try to understand, is this 8 colonies representative of the entire, you know, island, or is it, you know, are they, you know, the 8 sickest colonies in the entire island,
right,
or are they the healthiest? You know, like we really don't have an idea. So having better coverage about that, and knowing what you have, establishing your own baseline, because it very well might be different from what we have here in the continental US. So that's the first thing. The second thing is, because we can't do anything to treat or to knock down these viruses themselves, the only thing that can really be done is try to mitigate the vector, that is the varroa mite, that's spreading all of these different viruses and causing them to flare up. So good management practices to keep the varroa levels down will help keep those pathogens in check. And then the third thing is, to try and really work together because what you're doing with your bees, or not doing with your bees, is affecting your neighbor, right? So that, because the varroa can be spread, and therefore can the pathogens can be spread from one colony to another. So we really need to think about these things as population affecting our neighbors. So those are the three things that I would say.
Fantastic. Thank you so much. And that advice is absolutely amazing, and it's exactly, I think, what we also saw in the regional project, the need for greater hive management, better hive management, that is, greater communication and collaboration and togetherness amongst the beekeepers, and of course being able to understand exactly where we are contextually with respect to the diseases that we are seeing. So thank you very much, Dr. Tarpy. Next in our agenda, we have Mr. Gladstone Solomon. He is a master beekeeper and the owner of King Solomon's Apiaries. But one of the reasons why Gladstone was actually invited here is because Gladstone was part of that first finding of varroa in Trinidad and Tobago. He worked with Mr. Mohammed Halim and others in looking at the pest, because it was never thought to be in Trinidad, and Gladstone was part of that discovery locally. So Gladstone, what we would like to hear from you is that, having the experience, 30 years in beekeeping, and having also gone through this whole process of finding diseases in bees so many years ago, what advice do you have for beekeepers in terms of the way forward with respect to addressing exactly what David has indicated, addressing varroa, but also addressing the other viruses identified? So Gladstone, I'll hand over to you.
All right, pleasant good afternoon. Thanks for that introduction, Dr. Mahabir. Good evening, colleagues. Allow me a slight correction. I am not yet formally a master beekeeper, but I'm working towards it. So yeah, let me get straight in, but I think it's important to give a bit of a context before responding directly. How many of us know when varroa was first identified in Trinidad? All right, since I can't do a survey now, I'll just give you the answer. So varroa was first identified in Trinidad in July 1996, right? Do the maths, that's 20-something years ago. And roughly 4 years later, in August 2000, varroa was identified in Tobago. Now, the impact of varroa on both islands were significantly different. In the context of Trinidad, and I will speed up a bit here, let's say 8 years before varroa was identified, we had what I call milestones in the beekeeping history of Trinidad and Tobago. The most recent before that, the discovery of varroa is clearly what I would call a milestone based on its impact. But 8 years before that, we had the dismantling of the Apiaries unit and a dissipation of the role of extension officers and so on. That is on record as having an impact on beekeeping in Trinidad, more so than in Tobago. And going back further, 9 years before that, we had the arrival of the Africanized bees in 1979. The point I'm making, we had a series of events that led to a decline in the beekeeping industry, starting with the Africanized bees in 1979, the dismantling of the Apiaries, and then finally varroa. So that the response to the varroa outbreak in Trinidad in 1996 was against that background. And from reports at that time, and it's recorded in a paper that I did, there was a mixed fallout. Some beekeepers treated, some didn't treat, some swear that their bees were managing just as well. But the data suggests that after a couple of years, there was a leveling out in terms of the initial impact. So I'm saying that the initial impact wasn't as severe as, let's say, Tobago. And I'm moving over to Tobago now. Just recall that we still don't yet have Africanized bees in Trinidad, sorry, in Tobago, even though they have been in Trinidad since 1979. But yet we got Varroa 4 years after. So what's happening there? All right, we leave that alone. In Tobago, the impact, well, the Varroa certainly couldn't fly alone if Africanized bee couldn't make it, right? So we're wondering what's happening there. But that's just history. The impact of Varroa, of the identification in Varroa, and I'm not going to tell you where it was first identified, right? But it's the person who is speaking, even though I'm not telling you. In my apiary, and it was done by a group of visiting beekeepers from the UK. And they said, well, I start the story, Gladstone, I have Varroa. And I said, no, that's foul brood, that's bee louse. You know, many of you probably don't know the bee louse. But doctor, I think the Varroa has superseded the bee louse. Perhaps there is something you could answer later. I know they confirmed, no, they said, look, it's Varroa. We had 6 or 7 beekeepers visiting from the UK. Just before that, there's an interesting story, so I need to give it. Just before that, we had reports coming in from several beekeepers that, hey, something going on strange in my apiary. And those 6 beekeepers, we toured several apiaries on the island, and there was Varroa in every apiary. Every apiary. So that we recorded a 35% drop in the number of colonies within the first 3 months of arrival, right? And that's against a European bee. We imported some apistan strips, did some emergency treatment throughout the island. It leveled off, but roughly 3 years after, in November 2003, we had a more devastating outbreak of what we thought was European foul brood, but was later confirmed to be the parasitic mite syndrome. At that time, we lost 52% of the colonies. So beekeepers in Tobago had some serious losses, and our response was quite different to Trinidad to the outbreak of Varroa. We mass treated all the colonies on the island. The society at the time bought it. We were refunded eventually by the government. So that's it. That's just the background and the context. So now we have a mix, and in Tobago too, we had a significant drop in the number of beekeepers from the 40s to about 16 beekeepers as a result of Varroa. So against that background, what would be my recommendations? First, to all beekeepers, I'll say, listen, you do have a choice. There are what I call philosophies in beekeeping. There's the intensive beekeeping option, and there's the extensive beekeeping. There's the heavy commercial, and then there's the natural beekeeper. There's the Eastern style or the Western style, you know, the East-West conflict. It also applies in beekeeping, and their associated philosophies. Some beekeepers swear that they have never treated, they never will treat, and somehow they manage in bees, right? Others on the other side, I am one of those, and against advice of pretty experienced beekeepers who would have said, let nature take its course. You know, just watch your bees die, hope that 1 or 2 colonies survive and they breed from that. It was too heartbreaking having experienced that. So I was one of those who treated, and what I used initially was Apistan that was immediately available. It's unfortunately it's a synthetic, it's a chemical, but it was pretty effective. It in fact was responsible for salvaging most of the colonies on the island. But we soon migrated from Apistan to using Apigard, Apiva, no treatment methods, what we call the integrated pest management methods. Beekeepers use several options, but by and large, and because we had a very structured and productive society in Tobago, we whatever we purchased, we made it available to the beekeepers, whether they could have paid for it or not. All right, so beekeepers, my tip to you would be to be honest with yourself, to be honest with your beekeeping. It's your bees. We know that beekeeping could be done in several ways, but the science has established, beyond a shadow of a doubt, that the viruses associated with beekeeping impact production, impact the health of your bees. So that's a decision you have to make. I opted for the side of treating. I now use an organic compound. I've been using that for probably about 6 years, called oxalic acid. It's a vaporized application. There's lots of information on it online. I honestly have never done all these different tests, you know, the shake test and the sugar shake test and soak your bees in alcohol and count the number of bees. I know that that varroa is there. Once I begin to see varroa, then I know I am due, a treatment is due. One other thing, we have such a variation in the weather conditions locally. This year in particular, it's like the rain season of long time, lots of rain. Last year it wasn't so wet. And all these are factors that would influence the impact of the viruses on your bees. All right, so bear that in mind and do something that's in the interest of your beekeeping. I think I'll wrap there. Thank you very much.
Thank you, Gladstone, for that very very historical walkthrough. I mean, I learned quite a bit. I didn't realize Africanized bees were here since many years ago. It's very interesting, but most importantly, what is also interesting is that the option of treating for varroa has been open to beekeepers since 1996. But I have a question for you, Gladstone. You know, you said that at one point in time, there was a 35% drop in the number of colonies because of varroa, and that there have actually been some serious losses. You quoted parasitic mite syndrome, which we now know is related to varroa. And that resulted in a serious significant drop in the number of beekeepers in Tobago. I don't know if you've been communicating with your beekeeper colleagues in Trinidad. Would you be able to say, did the varroa have a bigger impact in Tobago as compared to Trinidad? Would you be able to say that?
Definitely bigger in Trinidad. And the current thinking is that the Africanized bees, which, you know, are the bees in Trinidad, are better able to cope with varroa. Is it, you know, it's said that one of the responses is the frequency of swarms, with which swarms occur among Africanized bees. Perhaps Dr. Tarpy could advise here, but I'm thinking that the subspecies, the European and Africanized bee, have variations in their tolerance levels to the mite and the resulting syndromes that come as a result from it. I think the Africanized bees, based on data I've seen and you know, chatting with beekeepers, I've done beekeeping on both islands. I know beekeepers very well on both islands. More beekeepers, very few beekeepers in Tobago, and come to think right now, I'm not too sure if any would tell me that they do not treat. In Trinidad, I know quite a few who would swear that they treat, that they never treated. Let me correct myself. There are very few beekeepers, if any, in Tobago, who I know who would say at this point in time that I do not treat my bees for varroa. Right, I'm just leaving it at that. In Trinidad, I know several beekeepers who tell me that they have never treated and they wouldn't, or they're not treating, right? There's a bigger truth somewhere inside there.
Okay, so in other words, you're basically saying, and that's something that we need to consider, that varroa is not limited to a specific strain or subspecies of apis or of honeybees. That if you think you could breed it out, by Africanizing your colonies. That's not going to work because Africanized bees are impacted by, are equally impacted by varroa as well as the other diseases that they carry, right?
But, yeah,
that's an important.
Yeah, the one I would perhaps give some consideration to whether they are equally impacted, right? I think that may have to be established. And I think we have a beautiful opportunity to do some kind of comparative analysis with the bees in Trinidad versus the bees in Tobago in terms of their response to varroa and, you know, varroa.
Sure.
Yeah.
Excellent. Thank you very much, Gladstone, for your
Most
input.
welcome.
I want to hand quickly over to Lena. Lena Dempewolf, as we said, is the biodiversity specialist at the Ministry of Planning and Development, but also Lena is an expert in pollinators. Lena, our question to you is, what are your concerns with respect to the possible impact of all of these new diseases, these 6 or 7 new diseases, to other types of pollinators that are in Trinidad and Tobago? Are you concerned about it?
Hi, Sharda. Good night, everyone. Thanks for having me. So my very quick answer to that is my biggest concern is all the things that we don't know. Pollinators are very understudied in the neotropics and the Caribbean and Trinidad and Tobago. We have very little information on a lot of these species that we have out there. We don't know what species we have. And as a consequence, we don't know what diseases they carry or what diseases they can transmit or what they're affected by. All we know globally is that there is a very significant decline in pollinators in native bee species, that our native bees are much better at pollinating our native plants and crops than the managed bees that we have, and that the diversity of bees is extremely important in maintaining that pollination supply. So what we need is not just a whole lot of bees, but we need a whole lot of different bees, because different types of bees pollinate different types of plants. Many of the honeybee viruses have actually been found in wild bees. In the handful of studies that there have been, most of those studies were in the United States and sort of northern latitude countries. So there hasn't really been anything done in our area, so we don't really know what we have. But from the few studies, and again, this is a very understudied area in wild bees in particular, some studies do suggest that the wild bees may have a slightly higher tolerance to these diseases, but that is only the species that they have looked at. So all of this I would take with a grain of salt and with a lot of caution. And the biggest thing for us to consider is just, you know, all the things that we don't know. That same study found that approximately 80% of the bees that they had sampled, the wild bees, all of them had shown or demonstrated some kind of virus, some kind of honeybee virus or the other. So as much as, if as much as 80% or more of the wild bees out there, and they were considering solitary bees, they weren't even considering social bees. So that means that transmission will have to take place where these bees meet, which is very likely at the flower. So there's a good chance that not only viruses are transmitted from honeybees to native pollinators, but the native pollinators may also act as a reservoir with these diseases that can then give it back to the honeybees. So it's really important to look at the whole thing as sort of a holistic picture. And it may not be enough to just manage honeybees to manage diseases, but you have to manage the others that they interact with, which are the wild pollinators. So you have to look at the pollination service on a whole that requires this, this level of management. And the other thing is that different species respond differently to these diseases. So while even, I think Gladstone touched on it a little bit in terms of different subspecies of honeybees may respond differently, but the differences are going to be much greater between different species of bees. So some may be greatly affected, some may be just a little bit affected, but also the effects may be different. It may not be that the entire species all of a sudden, you know, drops. It could be many sublethal issues. They could be flying just a little bit less, they could be a little less mobile, they could be a little more susceptible to being predated upon. Small things that add up to a huge problem in the grand scheme of things. So the biggest consideration again is that we just don't know what we have out there in terms of the diseases and in terms of how native bees respond to that. And we don't even know necessarily what species we actually even have. But because we have to think about this transmission at the flower, it's also very important to know which species of bees visit which flowers and which species of bees pollinate which flowers, and where these areas are of overlap where disease transmission can actually occur. A big concern is always that, especially with the importation of honeybees, or even if there were to be an importation of honey, the issue is that these new diseases can be brought in, which can not only be detrimental to the beekeepers and the beekeeping industry, but it could be detrimental to food production because it can wipe out populations of native pollinators that are required for crop production. So it has a much wider impact than that. And looking at where this interaction takes place and what the pollination network really looks like, who pollinates what flower, who goes where, that is very important. So we really also need to see how are the local species affected, not only who are the local species, but how are they affected. We are working a little bit on that in the sense that our BestNet CT project right now was working with the Trinidad Tobago Field Naturalist Club on a bee survey in Northeast Tobago for wild bees, in Northeast Trinidad and in Tobago for sampling wild bees to even identify what species we have. And we're also working on a plant pollinator catalog. We're trying to pull together all the existing data on the relationships between bees and other pollinators and the plants that they pollinate and what that sort of thing looks like. So we're trying to put together this catalog for many reasons. The other thing is, though, what also should be considered is that, are bees the only ones that carry these viruses? Are there other insects that can transmit them, that can maybe not be affected, but can pass it from one bee to another? Because there's a host of organisms that tends to go to these various flowers. So that's yet another consideration. So the problem is bigger than just at the colony. The problem can extend to the wider environment, and just having this additional information is really good. And it's great that we're, starting to get some kind of information on what is out there so that we can then better in the future manage this kind of stuff. So yes, I think my biggest takeaway is just, my biggest concern is that all the things that we don't know. And I'm trying to encourage people who are research oriented to maybe spend some time to look into that. Thanks.
Thanks, Lena. Thank you so much for that. Some very insightful information Lena has shared with us. Lena, I have a quick question for you. Now, Trinidad is blessed with a lot of stingless bees, solitary bees. We have a wide diversity in terms of our insect population. Just generally in our biodiversity, we're probably one of the richest in the entire of the Caribbean. But many of the other islands do not have, just like us, don't have a survey of that biodiversity. They don't, some of them don't even know their bees as well. And many of them claim that they don't have local bees, or we don't have solitary, or we don't have, we don't have stingless bees, okay? And I'm saying this because when we did the beekeeping survey, that's exactly what the beekeepers told us. We have honeybees, but we don't have others. As a lot of these countries are looking for alternative methods of sustaining themselves economically, agriculturally, etc., they may be importing more honeybees, but not necessarily any other species. Can you explain to us whether or not there are other types of bees that have some sort of economic value and what that potential economic value could be?
Okay, great. So they're immensely important. Pollinators, so in terms of bees, the majority of the bees is actually not social. Only a very small proportion of bees make honey and are social and live together and so on. So honeybees is one set. The others that we have on some Caribbean islands, but not all, are various groups of stingless bees. And those also produce honey in much smaller quantities that has mostly medicinal value that people use. And the vast majority of bees are actually solitary, so that means they're on their own. And they are, the vast majority, again, is ground nesting. So they nest in holes in the ground. There are others that nest in small cavities in wood and things like that. Others roll leaves. So there are lots of different species of bees that have lots of different habits. And their really big importance is in, first of all, in agriculture and food production. Because even though, I've heard a lot of people say, oh, I will just, take a paintbrush and I will do the thing. First of all, that takes a lot of time. You really have to know what you're doing. It's very finicky work, and it has been shown that it's actually not nearly as effective as when you have bee pollination going on. With bee pollination, the fruits and whatever else you're getting out of it is of a higher quality and greater abundance by far when native bees pollinate them. And it depends. Some pollinators and some native bees visit many different types of plants, and some only visit one specific species of plant. So if you have one that is very specific and the pollinator disappears, then the plant also disappears. So there's a big issue in terms of the diversity being reduced, and the that also having an impact. And additionally, the whole concept of ecosystem services and people being just being out in nature and enjoying nature. And you enjoy the nature because of the diversity that you have. You're seeing the waterfall, you're seeing the different plants, different animals. All that is based on pollination, because without the pollination, you will not have the plant diversity, you will not have the soil retention, the water filtration. So pollination underpins all those things. So economically and socially and culturally, it's extremely important.
Right.
And in addition to that, many of the bees are very different colors. So people may not know, people may think they're flies actually, because they may be black or blue or green, have different shapes and sizes. So education is really important. And I would honestly encourage any island to be looking into doing some sort of bee survey so they can get a better idea of what they have.
Right. So let me turn the question around and ask you this way. Traditionally, people have imported in some of these smaller islands of honeybees. Isn't there an opportunity to also import stingless bees and sort of develop that stingless bee keeping sort of in order to be able to how should I put it, be able to build that industry, thereby facilitating or continuing the diversity of these creatures on the different islands?
That's a bit of a tricky question. So I would say hypothetically, yes. From an ecological perspective, I would be careful because you don't know what the impacts are going to be.
Okay.
So in many cases, these bees have already moved into places where they haven't been before. So they occur naturally. But you are not really quite sure how they interact with the other local species. Are they going to drive out the local species of their habitat? Are they going to outcompete them? Are they going to cause problems for the local wildlife? But yeah, but also again on the issue of disease, because again, we don't know yet who transmits what. So there are lots of things that, I mean, hypothetically, yes, they could be treated just as honeybees. They might actually be easier to manage because they are smaller, they don't fly nearly as far, and the queens cannot swarm. So that hive that is that is built, they stay there, and the queens are replaced, and it can go on for 100 years if anything. And when they do move out and move away, and the new queens find new places to go, there is a period of exchange between the two hives where they go back and forth. So you have a better idea of managing it and controlling where they go for sure. But I would still say that some more information is needed in terms of diseases, impact on the ecology, and so on the islands.
Right. One quick question. I mean, we have 89, almost 90 people on this call from across the Caribbean region. Can they help you with the data collection in terms of what insects pollinate what flowers? Is that something that can be done?
Yes, please. So we have set up for every Caribbean island on iNaturalist, and iNaturalist is a citizen science program platform that you can, it's an app you can download on your phone. If you prefer to go out with a fancy camera and upload pictures through a website, you can do that as well. And you can go take pictures and upload it, and then somebody will identify online, the experts that will help identify what you're seeing. On there, you can set up projects. If you wanted to, you could set up your own project for your own area. But I've already made your job easier for you because I have set up one project for every Caribbean island that is called Pollinators and Pests of, and then the Caribbean islands. So what you do is you join the project, and when you post your pictures, you tag that project, and then you're automatically collecting this data for your island. The nice thing is that anybody can download it. There is a button where you can get a CSV file with all of the information for the project. It is very useful for research purposes or in general. You have control over the privacy of where you're uploading the picture. So do you want other people to see where the location is or not? You can adjust that accordingly. There are lots of things. And if you want to learn more about iNaturalist, I'm happy to help you out with that. The reason why it's called Pollinators and Pests of Trinidad and Tobago, or Pollinators and Pests of Antigua, or Pollinators and Pests of Jamaica, or whichever island, is that one, we'd also like to be able to track agricultural pests and see what the problems are, what people are having problems with, so that we can see what are more targeted responses in terms of pesticide use and treatments that are not going to wipe out native pollinators. And also because sometimes they're very complicated relationships between pollinators and plants. For instance, a caterpillar by a farmer might be considered a pest, but the butterfly is the pollinator. So these are two versions of the same organism that have two different roles in the environment. So we wanted to capture all of it. But I'm happy to help anybody out, do sessions with people, school sessions with organizations, anything. Let me know, I'm there to help.
Excellent. Thank you very much, Lena. And of course, it's been a very exciting discussion. Thank you so much for your contribution. We want to continue discussion by calling in Ryan Rawlins quickly. Ryan Rawlins is the director of Rawlins Bees. He is an avid beekeeper, young, exciting. When Ryan talks about his bees, he's passionate about them. He loves his ladies, as he says. So I'm going to hand over to Ryan. Ryan, are you there?
Yes, Sharda, I'm here.
Fantastic. So, Ryan, I have a question for you. So basically, we've sat down in this discussion so far. We've heard that varroa mite is a major issue in Trinidad and Tobago and has been that way for a very long time. That varroa is a vector for plenty diseases, viruses of honeybees, and that it's very prolific. We've had so many indications that the varroa and its associated diseases are causing so much problems with bees. You're a young beekeeper. Are you in any way daunted by this, first of all, this information? And secondly, what would you do, or what would you say to motivate other young beekeepers, to keep going, just to keep, at the work and, not giving up?
Yeah, okay. Well, daunted is a kind of vague term, but it's more like unbelievable because the amount of viruses we found on the samples alone. Because I could say with my colleagues and so forth, we don't know 90% of the viruses that came from varroa mite. And the problem we're having is most beekeepers in Trinidad, as Mr. Solomon said, is that they don't like to treat for varroa mite because we like to say how the Africanized bees, I don't want to say resistant, but tolerant of the varroa mite, but yet they're still present. And what they're saying is that they'll let it swarm, and some beekeepers don't treat for mites and some do. So the encouragement I would say to the upcoming beekeepers and them, you need to treat for your mites, do your mite counts, keep on trying at the end of the day. Once you know you have varroa mite, you treat for it, you'll control it, and then your chances of having those viruses are much lower, and then you'll have a much more powerful hive, and therefore it will directly affect your honey production.
Right. Okay, so let me ask you something. In terms of you said do your mite counts and keep treating. Now, as a young beekeeper, Dr. Tarpy said something that was very important, that as neighbors and as fellow beekeepers, we have to be cognizant of our work. Do you see that as being important as a young beekeeper, that there's a need for greater communication as well as working together among beekeepers?
Oh, most definitely. You know,
Okay.
if I treat for varroa mites and my neighbor, who's also a beekeeper, doesn't treat for varroa mites, there will still be the contact at some point at that flower point. What Liana said, that the varroa mite could transfer from one bee to the other, and therefore the virus will still be in my apiary. So it's I truly believe that every beekeeper in Trinidad and Tobago should be treating for varroa mites.
Okay. And okay, so if we are going to do that, do you see the importance of persons coming together? Because I mean, aside from the fact that you need to treat independently so that we kind of control the diseases in your individual spaces, so you kind of expand your treatment and control of diseases in your communities. But you also see the value of communicating with your colleagues, your friends, your close
Yes.
by beekeepers, because I mean, collectively, if you were to purchase, let's say, treatments, you would be
Yeah.
able to get a better price for it if you were to work together.
Of course, wholesale price is always cheaper at the end of the day, so it'll be less on your pocket. So it's just nothing to have a coalition of beekeepers to buy to acquire the mite treatments, and at the end of the day, it won't be detrimental to your pocket. You know.
Mm-hmm. Excellent. So Ryan, thank you very much. In the interest of time, let me move on quickly to Richard.
No problem.
All right. Appreciate your feedback and appreciate your enthusiasm. And now I'll hand over to Richard Matthias. Richard, are you there?
Yes, thank you, Dr. Mahabir. I am here in present.
All right. So I'm putting up your presentation for you. Can you see it?
Yes, thanks. You could put it in full
presentation mode. Right. That's correct.
Yes.
All
Thank
right.
you.
So Richard.
All right. I'd just like to say welcome and thank everybody for joining us this evening. We've got quite a turnout, which is great. And I think some of the information has been shared by Dr. Tarpy, Dr. Dempewolf, Dr. Mahabir, Mr. Solomon, and Ryan has been very revealing, and it really plays a part, shows us that we really need to play a part in apiculture and trying to improve our habits. And what we're really trying to do in this what we're doing today is to shine a light on what's happening in particularly Trinidad and across the region. So in my presentation, I'm really going to talk a little bit about what we know to date and how we can move forward. So if you go to the next slide, please, Dr. Mahabir, be grateful. Okay, so as we had discussed earlier, data collection is key for us to really improve the information and look at some of the information. In order to understand the Caribbean context, a review was completed on the distribution of varroa and other viral diseases. Data we used for our review came from various ACBO Congress papers, some scientific research papers, our regional beekeeping survey, which is done part of this, the Jeff SGP South-South collaboration, and also in-person surveys and field work in the field, observations from in the field. Next slide, please. So in our review, what we know for certain, both scientifically and anecdotally, is that varroa mite is present in virtually all of the English-speaking Caribbean islands, except for Barbuda. I've been to Barbuda with team from Barbuda, and I've tested virtually, I think, every single beehive in Barbuda, and we found no varroa mites on the island of Barbuda. So I think this is the last bastion in the Caribbean to be varroa mite free. So there is one country in the world, Dr. Tarpy, that still is varroa mite free, and that's Barbuda, in a little island in Barbuda in the Caribbean. That we still think is varroa mite free. But anyway, let's push on. There's a need to collect data from the islands not included in the map. There is also little or no work in some countries on the diseases distributed by varroa. As Dr. Dempewolf suggested, spillover from your managed colonies of bees to infect native pollinators is something that urgently needs to be looked at. And then you also have the reverse in spillback. Feral bees reinfected your managing colonies. So, you know, even if you treat your colonies of bees, and feral hives in the environment can still have varroa and bring it back to your bees. So we really, really need to look at things in a holistic standpoint. I really try to do our best in all of our islands to manage varroa populations. And as has been explained by Dr. Tarpy, these are a list of the viruses that were found in Trinidad and Tobago in the first column. We also were able to pull some comparative data from Guyana. There's been a few studies that have been done on viral loads in Guyana, and we can see on this chart what viruses were found there. We were able to pull up some scientific papers from Puerto Rico and also Dominica, where high levels of nauseum were found in Dominica at this time, and you've got a whole host of viruses that are present in North America. So if you look at it in a context that to our south of us, we know what's present in Trinidad and Guyana, and to the north of us, we know what's present in North America and somewhat in Puerto Rico, and a little bit of information on Dominica. We have results pending from St. Lucia, which are still under review. And we know that virtually every single island up the Caribbean island chain has varroa bar Barbuda. So it's, you know, anecdotally, you're probably going to find all these viruses or a combination of all of these viruses present in over all of your islands without us even having gone there, taken samples and sent it up to North America to Dr. Tarpy for testing. So, you know, I would strongly advise, if to your south of you you've got a bundle of problems, and to the north of you has got a bundle of problems, and you're right in them slap bang in the middle of that, you probably got problems too. You just don't know it as yet. So I would strongly suggest that beekeepers, based on the anecdotal and the scientific information that's out there, I think most of us have seen deformed wing virus in some, or if not all of our colonies across the Caribbean. And that's one of the few viruses that we can see with the naked eye. So I think, you know, it's very important that we treat with, you know, in the absence of definitive proof that all these viruses are there, I think we've got enough anecdotal evidence that we should treat. Can we move to the next slide, please? So can these diseases spread in honey? Yes, some of them can. Spores from Nosema apis can be transported in honey, and American foul brood and European foul brood. They can be found in the honey and pollen. So, you know, we really have to be cognizant of the diseases that are present and the diseases we're unaware of. And I know there's a lot of discussion currently in Trinidad and Tobago in the opening of the market for imported honey. And, you know, this could be problematic, I think, for Trinidad and Tobago. And I think, you know, all the beekeepers in Trinidad really need to rally around together and make their voices heard and use some of these scientific evidence that we've brought to bear as to reasons why your market should not be opened, or if it is, it needs to be very, very, very heavily regulated. Otherwise, you know, it could be like really bad. And I really, you know, I've got a lot of friends in Trinidad. I know a lot of young beekeepers are aspiring into the industry, and I think it would be very disappointing to them if the wheels were taken off their apiculture train, so to speak, before they even left the station. Next slide, please. So I think this map is even more interesting and makes my point even clearer. If restrictions on the importation of honey are lifted in any of the Caribbean countries, there could be regional implications. Allowing the importation of honey can spread diseases already identified in the region, as well as introduce new diseases like American foul brood and European foul brood. As such, any importation allowances must have strict restrictions and criteria to safeguard local biodiversity from potential diseases. And I think, as you can see from this map on the right, we know that there have been issues with American foul brood and European foul brood in South America, and this can easily be brought up through the different countries up into Trinidad. And if you open your markets to international trade, you could possibly have honeys coming in from North America, bringing in a whole host of viruses as well. And then because of Trinidad's high industrial output, and it's really a transshipment hub to the rest of the Caribbean, potentially bees that are infected in Trinidad could find their way up the island chain and bring these diseases along with them. And that's depicted in the 2 red arrows. So it's something that, we really need to look at. I know there is some moves afoot at the CARICOM level of trying to, allow regional trade of honey. However, this discussion seems to be happening amongst technocrats, and no beekeeping organizations are part of the discussion. So, it's really very important that as a community, as beekeepers, our voices are heard as a single voice and very loudly. And we really need to protect the markets with each of our individual countries, and especially as a region as a whole. If we can work together to find some way of disease monitoring amongst all the Caribbean islands, fantastic. And if there's beekeeping organizations or individual farmers that are under some kind of type of regulatory framework that guarantees that their honey comes out clean, fantastic. And this can be moved around the islands, fantastic. I'm all for that. But an unregulated environment where honey moves freely around the Caribbean, I think that could have catastrophic effects for us as beekeepers. Next slide, please. So as Ryan suggested, and as Gladstone suggested, integrated pest management is the key. Management for varromites. How can we stop these viruses? Very easy. Stop the vector, knock it down very early, and have an integrated pest management plan for your diseases. This is the best way to control it. Try to breed, or if we can create a breeding program of varroa-sensitive hygiene queens, that would be a plus. If we can do regular mite load testing, use miticide treatment, I recommend Ulencap, and Ulencap is spelled A-L-U-E-N, not A-U-L-E-N, and supplemental feeding to boost your bees' diets at certain times of the year when there is not much around there. Bees need a collection of amino acids for them to live successful, healthy lives. And at some times of the year, we as beekeepers need to provide these nutritional supplements to keep our bees going if we're going to keep them in good tip-top shape. Next slide, please. So, recommendations. There is a need for further resources to be developed to answer the following questions. What is the distribution of viruses in the Caribbean islands with varroa? Are any of these diseases being detected in honey from these islands? This is research that needs to be done. Are the diseases impacting other pollinators? You've got spillover situations where I think in Europe and North America, they found deformed wing virus in a number of bumblebees. All right, now if the bumblebees catch the varroa catch or contract deformed wing virus, you're treating your bees, they can quite easily bring back the deformed wing virus back to your bees. So, it's very important that we really start looking at all the pollinators in our environment and try to take a holistic approach before we really have some serious problems. And how can we encourage persons to implement an integrated pest management program for these diseases? It's very important as beekeepers for you to be reviewing your mite loads in your bees. You may be very reluctant to treat your bees for varroa mite, but at least check to see what's there. Check and see if your bees have varroa mite, and then make a decision whether or not are you going to ride it out or you're going to treat. I've done testing for varroa mites in Trinidad, and I've found some very interesting results. So I would recommend all beekeepers across the Caribbean, don't throw the concept of treating your bees totally out of the window. At least test to see what your mite loads are, and then make a decision based upon your mite load, whether or not you're gonna treat or not treat. And that's my presentation, very short and sweet. Anybody has any questions? I think Dr. Mahabir, we should open the floor. I'm sure everybody's should be chomping at the bit.
Yes,
to ask a few questions.
I agree. I think this is an excellent time to stop, because we've brought up a lot of interesting points this afternoon. Just very quickly to summarize, varroa is one of the main vectors for many of the viral diseases that we saw today. Richard is saying that those diseases are being detected in quantities in Guyana as well as in the US. That these diseases, the best way to address them is treatment. And then there has been some success in terms of management of the disease in Tobago, because beekeepers are very good at treating in Tobago. The treatment is important not just for your colonies, but also for the pollinators that survive around, and also for the management of the sector, to ensure that we could continue producing honey locally in Trinidad and Tobago. So I'm going to pause there. That is my attempt at trying to summarize quickly at least one succinct point from each of the presenters. I'm going to open the floor now to questions. Are there anybody in the team, in our is there anybody who has any questions? I see immediately Alan Williams. Alan, feel free to unmute and ask a question.
Okay, good morning, good night, everyone. The information was very vital. I'm very glad to be here. There are two. First, I just want to make an observation, because my name is Alan Williams. I'm from St. Vincent and Grenadines. And what we have seen with the varroa mite, we are a multi-island state, and when it was introduced, well, when we discovered the varroa mite in 1998, and by that time, the smaller islands, it was completely decimated. All hives were actually gone. And we were in about 2006 when we had about 7 or 8 remaining colonies, and those colonies, we did a queen rearing program that we were able to rebound. And we also had imported hives from St. Lucia, and we were able to revitalize the industry. My question is, it might be a long shot or recommendation, if we can try to improve the plant diversity within the radius of the bees or the vegetation by trying to plant a greater diversity of crops. I think if we are able to achieve that, that might be a long shot, but that can be another way of organically giving the bees a variety of nectar sources and food sources, so that they can be able to build a resistance against the various viruses in conjunction with treatment, right? And also, because we have two different types of climate in our islands, where there is less rainfall and dry areas, we see the mite is dwindling. The bees have a higher tolerance level, but in areas that is high in moisture content in the atmosphere, we see the bees have a greater challenge in controlling the varroa mite. So you will see the production of honey much better in the dry island, which is the south of St. Vincent and Grenadines, and on the mainland, where there is more rainfall, we see hives are actually struggling to develop the resistance for the varroa mite problem. What we have seen too as well, the bees that survive the varroa mite, they naturally devour the varroa mite with the mandibles, and they also control the drone population during the year. So only just between the peak areas of the year, you will see drones, and during the most time of the year, you can actually count the number of drones on your fingers that is in the hive, so that the bees themselves also have a natural control on the control of varroa mite. And also, that's my contribution. I know we have to try our best together to work with the bees to see how much we can help each other and make our livelihood a success. So I would encourage other young people to look forward to a very dynamic way of entering your beekeeping experience. Thank you very much.
Alan, invite us to the Congress. Don't forget.
Oh, yes. Well, of course, St. Vincent and Grenadines will be hosting the Congress, and we have much to talk about. So looking forward to see many of you to join the Congress. We're now having it on the 30th of October to the 2nd of November, that weekend there. And we're looking forward to see you, and we will have a good time together in St. Vincent and the Grenadines. Thank you.
Thank you. Thank you, Alan. We have Nikosi. You're next. Nikosi, unmute and you can go ahead and share.
Okay, good night. I'm good night to everyone, and thanks to the presenters. The last Alan's raised a few points there that got me thinking. So excuse me if I'm developing my question while speaking.
Mm-hmm.
He spoke about planting flora that will help the bees. And
trees for bees, bees for trees, that's correct.
Right, yeah, but I'm thinking it should be, especially for native bees, native flora. But my question, what I'm thinking about is, when it comes to resistance to the diseases, has there been any research on the natural flora's ability to help the bees resist the viruses? Because we know the varroa itself exacerbates the issue. But what about the natural flora and the multitude of minerals and vitamins that the bees will forage on? Is there any research that can help us use, besides obviously the treatment for varroa, but help bees resist the viruses also?
So I'm going to hand over to David. David, you can take that question.
Yeah, I try to handle that one. That's a difficult question to answer. Mm-hmm. The answer is no, simply because it is so highly complex. But in essence, I don't think that there's going to be kind of some magic pollen or nectar out there that's going to kind of right. cleanse the bees of these viruses. But I think there's a broader point that good forage, well-fed, healthy bees have higher immune systems right. that are much better able to withstand. So I don't think it's kind of one specific magic bullet, perhaps, but there is kind of an overall nutrition, and I think Lena had touched on this as well, and might right. want to chime in here, that kind of better forage, less stressed bees, the pathogens don't have the same opportunity to take a foothold. So, trying to make sure that the bees are not nutritionally deprived is the best answer, not necessarily one plant over another.
Yeah, for sure.
Right. So I think that's an excellent answer. I don't know if Lena, you want to add anything to that response?
Yeah, no, I totally agree. I would say that, just like with people, if you have a healthy diet, you're less likely to get sick. So there's, and there's, again, in terms of, excuse me, plant compounds, there's no magic bullet and there's no magic pollen and there's no magic nectar. And we also don't really know in terms of research what is out there, but just having a wide variety of native food sources is always a very good start to sort of keep away diseases.
So this also, excuse me, ties into the larger discussion, I guess, on preservation of the natural environment and the natural,
That's correct.
natural areas, right? Because the reason I'm thinking about that too is because when I've been called out to remove wild hives, especially in urban areas, it seems there that the bees, at least in my just observation, no science behind it, they seem to be doing quite well. And it's also, I think it's because of the diversity of the flora, as opposed to a forest where they'll have some trees tending to dominate. And the bees there, the wild hives in urban areas tend to seem quite strong and quite, and that was a surprise to me, so.
Well, there's actually some scientific publications to back that up.
Okay.
That diversity of different pollen sources is better than kind of one main source.
Right.
But again, there's a lot of devils in the details. I actually do want to chime in that there has been some work done on one specific pollen type, and that is sunflower pollen, that has been shown to cleanse honeybees of nosema in their guts and bumblebees of their kind of gut parasites as well. Even though it's not necessarily the most nutritious pollen, the spiky, pollen grains themselves has been shown to kind of cleanse bees of certain diseases. So I wouldn't call it a magic bullet or something to rely on, and it doesn't really address the viruses per se.
I see it.
But there are examples out there of how there is kind of different plants that could be used medicinally by the bees themselves, which is interesting.
So I just want to that's really interesting, because if it is that the shape of the pollen grain is what it is, then I'm sure that there must be native equivalents to that, and that's where the shape of pollen grains becomes important, and more work should go into something. We should really look into how our native pollen grains can have a similar function. So I think that's something to flag for sure.
Yeah, and I just want to put a plug in here, following from what was said. Part of Lena's BestNet project is to create a pollination catalog, where she's actually going to be looking at these kind of things. What bees are going on what flowers, but does that flower have a specific shape or size of pollen that attracts the bees, that keeps the bees, say whatever it is. And the thing about it is that to do this research, and this is where I'm plugging for Lena's work on BestNet at this point in time, what Lena's trying to do is research that if one scientist would start in one country, will get nowhere in a very long time. But this is why the iNaturalist platform really is so important, because it offers the opportunity for citizen science. Any secondary school student can now go into doing something, a little bit of work like this. It's not, we're not asking you to write a PhD. We're just asking you to do some preliminary work, so we know where to highlight or where needs to be highlighted in certain terms of continued work. So that's where we have a lot of questions. Harvey, you want to go ahead and unmute and ask your question?
Okay, good night all. My question, it's kind of taking us a little off the main area of viral load, but it has to do with the stingless bees. I can't remember whether or not it came out in the presentation, the levels of resistance and if they're faring better than the stinging bees. But my other concern about that stingless bees, in terms of introducing it in anywhere that it may not be present at this time, is the whole issue of predialasny. We know that is a serious problem in any form of agriculture. And for many, many years, because of the fact that bees sting, it has been a deterrent. But we are now seeing even that, that armor that we had before is now being lessened, and trying to introduce stingless bees around this time, I have a fear that it might not work out in our best interest. Just my comment.
Oh, sorry. Okay, great. Thank you. Joshua, you want to go ahead and unmute?
Sure thing. Pleasant good night to everyone on the call, all panelists, and the audience at large. I caught wind of a project that happened or is happening in Singapore, where researchers have been breeding a type of mosquito that essentially either hinders fertility of naturally occurring mosquitoes, as the case may be. So it's a sort of vector control program. I wondered if any of the panelists, first of all, heard about such a thing being done, as well as what might be the ecological implications of such a thing being done with the varroa mite.
So I'll go ahead and take that. Actually, there are several different programs going on that are trying to use genetic engineering to make Aedes aegypti, the malaria vectoring mosquito, incompetent in being able to vector the malaria parasite. And then kind of release that into an area, and then malaria rates can go way down. And with 1 million deaths per year of malaria, that is a very important thing. No such thing is actively being done or even proposed to be done in honeybees. That is, there's nobody that I know of that is trying to genetically engineer honeybees and trying to into something that they aren't naturally. But there are plenty of breeding programs that is taking the natural that is in the bees right now and trying to selectively favor different types of bees that are of varroa or more tolerant to are just going to naturally have a predisposition to having lower levels of virus. So there are those breeding programs underway, but I think that really is something that is going to be regional and country specific, because we don't want to be shipping different genetics of bees and their potential problems across national borders. So I think there is a great potential for that, for something in the Caribbean region, for local selection of stock for traits that are favorable to your neck of the woods rather than here in North Carolina versus Canada, right? Those are all very different places where different traits are going to be favored. And there is enough genetic variation out there where one could selectively breed for tolerant stocks, for sure, but no genetic engineering like in that mosquito example that you had brought up.
Understood. Thank you very, very much.
Are there any other questions from any other participants? Let me just check the chat. So Lena's been sharing her iNaturalist project on the chat. I also shared a video on how to test for varroa mite. And what I would like to do is throw out a challenge today. So we've had at least 2 to 3 of the actual presenters this evening speaking about the fact that we need to have good management practices with respect to hives in order to be able to control varroa, which we are going to assume is a major vector for a lot of diseases that we're seeing in Trinidad right now, in honeybees, that is. So we're talking about good management practices. And, one of the things that I have always, that I strongly believe in is citizen science. So just quickly, oh gosh, if I have a minute, I hope I could do it in less. My background is actually in environmental biology. I'm a water quality specialist, strangely enough. And one of the first things that I did when I left the university was create a citizen science water quality testing program. And that is actually one of the first in Trinidad and Tobago, and I think probably in the Caribbean. The whole idea is I love the idea of persons from any walk of life. I love the idea of breaking down the science, first of all, and allowing persons from any walk of life to be able to make a change and contribute to something that is very important in terms of solving a problem. Here we have a problem of diseases in honeybees and potentially in other pollinators, and we have very little information. So far, we've been able to give you some advice in this presentation on how you can address some of these things. So we've had wonderful advice from Dr. Tarpy, which is basically that we need to do more surveys of diseases that we're seeing. So we want to do a little more of that survey information. We also, he also recommended that we have good management practices, but that has also we work together as persons in interest in insect pollinators and bees, that we work together in order to be able to make a difference. So I'm throwing out a challenge today. I've put up the video on how to test bromide. And my challenge that I would like to develop through you as all of our partners here is to set up a Citizen Science monitoring program for varroamite. Now, varroamite is very, very easy to at least test. You need a varroa tester. You can look at the video. A simple varroa tester, a little bit of alcohol. You scoop some bees up from your apiary, you shake them. And what happens is that because of the sieve that is located in the actual varroa tester, these varroamite fall off and you're able to count the number of varroamites. And there's a limit, it's an acceptable limit of 2 to 3 varroamites for every 100 bees. Now I'm telling you all of this because I'm challenging the people and what 8 or 9 countries that we have here today. Let us see how we can start a varroa monitoring program for the entire of the Caribbean. And all it would mean, it costs, it literally is what, 20 US per country, or anything, or per organization. They're representatives from beekeeping organizations here. You need 1 varroa tester. And you need a little bit of alcohol, which we all know we have because we're still sanitizing our hands from COVID. So if it is that we're able to do that, I would challenge us. And I mean, I could work with Lena. We could maybe add a little link to the iNaturalist where somebody could enter like a Google form, and you could say what country you're in, what part of your country you're from, and then where you tested for the varroamite and how many varroamite you got. Let's start collecting that baseline data, because that's the kind of data that is going to help us to make a case for being able to access funding to do this genetic testing in all of our countries for the kind of diseases that varroa may carry. So that would be one recommendation that I would definitely want to make out of this. The second thing I would want to also recommend is how can we now collectively across the region implement integrated pest management. Now we've had a lot of recommendations as to what that integrated pest management involves. We've had, for example, proper hive management, so checking on your hives regularly. That was mentioned during the, I think Ryan mentioned it. We also had Gladstone saying that regular treatments of your hives is a very important aspect of the hive management. We had Richard talking about actually testing regularly to make sure that your varroa levels are very low, as well as queen rearing. We had 2 persons of the team this evening talking about queen rearing, and that is opening up another big conversation about how we can get more queen rearing and more relevant queen rearing programs in our countries in order to address some of these diseases. So I'm going to hand over. I see Linda has put up a hand to ask a question. I'm going to hand over there, but I've thrown out a challenge and some food for thought. And I would like to see in the chat all of those of you all who might be interested in participating in a project of that kind. Lyndon, you want to go ahead and ask a question? You can unmute. Lyndon Stewart, can you hear me?
Hi, good evening, everyone.
Sure.
Good evening. This is Lyndon Stewart from Georgetown, Guyana. And I would just like to ask a question. Do you still have a large population of Africanized bees in Trinidad?
Yes, yes. Most of our bees are actually Africanized.
Yeah. And you're getting problems with varroa mites and the Africanized bees over there?
Yes, actually, it's interesting that you should say that and ask that question. But yes, the varroa mite is present on the actual Africanized bees, because there's a misconception that Africanized bees cannot have or more adapted to caring for and removing varroa from their hives. That's not true. And I think I'm going to hand over to Richard. Richard, maybe you might be able to give some experience from Guyana, having worked in Guyana and tested in Guyana for varroa mite. Richard?
All right. Well, that's why I asked that question. All right. I joined the meeting late. Sorry about that. I joined the meeting late, just about a half an hour ago. But our first diagnostic test was done in 2011, and Mr. Gladstone Solomon, along with Clive DeBrain, was here in Guyana. And varroa mites were one of the things that we were checking for. Then our second diagnostic test in Guyana was done between 2013 to 2014. And our third diagnostic test for pests and diseases, including varroa mite, was done in February of 2020. And Guyana is Africanized bees in all 10 of our region that I've traveled to do training and research on the different species of bees. And from 2011 to now, we are doing checks within 7 of the regions, because not all the regions are easy accessible to get to easily. Some of them you have to get there by aircraft. But the 7 regions that we are doing checks, between 2011 to now, we do not have an alarming rate of varroa mite. Matter of fact, in 2020, within the 7 regions that we have traveled, we found 5 varroa mites. In 2013, the same thing. And 2011, I think around the same thing. And Mr. Solomon can testify to that because he was with us. So why I ask if the Africanized bees in Trinidad is being affected heavily by the varroa mite, it's kind of leaving me worried now that someday we might be experiencing a high infection of varroa mite. But so far, for now, as they say, we got enough wood, we have not having any problems with varroa mite infestation in any of our hives. Matter of fact, we check for tracheal, we check for small hive beetle, and we have never seen those things in our hives. So I don't know if it's the different type of climate, but I think Guyana and Trinidad has the same type of climate. I don't know what was the problem, but we do not have any species of Italian bees in Guyana anymore. Early this year, we also had the farmer-to-farmer program sent in professor from the States to come to do a test to see if there are any disease that can be transferred from the melliponas to the Africanized, or if any can transfer from the Africanized to the melliponas. We are still waiting on the results on the melliponas that they collect samples from. And there were some melliponas that we found that we did not even knew that they were in Guyana. We were only knowing about it for Fosa, the Trinitatis, but now they found one called the Rawlin. So it is shocking to hear that you're having the problem with varroa mites on the Africanized bees. What other problem you're getting with the Africanized bees over there? Are you seeing any small hive beetles in Trinidad in the hives?
Hold a second. Richard, you might want to take that question.
I'm just, okay, so I don't think there's any issues with small hive beetles, Lyndon, but I'm actually looking at your 2013 results for varroa mite, tracheal mites, and nauseum results that were presented at the ACBO Congress. All right, the majority of your hives, of the 26 colonies, I think that were tested. I think you did have some colonies that showed up at 18 mites per 100, 12 mites per 100, just under 9 mites per 100, you've got a 6, and you've got an 11. So I wouldn't say you've got a 0 mite problem in Trinidad, because I'm actually looking at your results. They're posted on the ACBO website, which would suggest contrary to what you're saying. I say that respectfully. But I think I've done some testing up in Berbice the last time I was in Guyana. I didn't see anything alarming. I mean, I saw like a 6 and an 8 mites per 100, but that's still above the threshold. When you're looking at a threshold of 2 to 3 mites per 100, anything above that is considered problematic. So, I mean, I think for in the case in Ghana, I think, you know, more regular mite testing by individual beekeepers and correlating of that data on a more regular basis might be required. That's my personal opinion.
Do you have our 2020 report?
I'm looking at in front of me right now. I was looking at your I've got your 2013, and I'm looking at your 2020 results now. And your 2020 results, yeah, I found your 2020 results were unusually low compared to your 2013 results. And I looked at that, and I found it. I found the data unusual, slightly odd, but the data is the data. I found that the data was quite low. The results as compared to 2013 were like very, very like almost a 180-degree turn, all right, as compared to the 2013 results. So I think I'll be interested to see what your current data looks like from all the regions. I'll be very interested in that, because I was in Ghana. I was in Ghana in I think April of this year. I think I tested a couple of hives in Berbice. I found a 6 and an 8 mite count. And that was April of this year.
Which area in Berbice?
I don't want to name the beekeeper's name. That would not be nice. He's not here.
No, because we did test in Berbice. We did in Fort Wellington. We did in Sanford.
Fort Wellington. Okay, Fort Wellington. So we both know.
Yeah.
Okay.
Yeah.
So in Fort Wellington. All right. So I think time of year is also very important for when you do your mite testing. And were these colonies treated prior? I mean, there's no data. The information doesn't state much if they were treated recently or not treated. So the information. There's some pieces in the puzzle that are missing in the Guyana survey.
Guyanese beekeepers do not treat for anything. They don't know anything about treatment. Anyhow, is there a certain season or a certain time of the year when varroa mite is more rampant in the hive? Is it during
it'll be in the peak of your honey flow. I mean, you'll start to see the mite populations grow as the honey flow continues, and as your honey flow decreases, your mite population will stay high, and probably you want to test at the beginning of your season, in the middle of your season, and at the end of the season. These will probably be the best time. You should really test 3 times a year to really gauge where your colonies stand. Just testing once a year, I don't think that's really enough data. You really want to test it right in the middle of the honey flow, and at the end of the honey flow, and then you would get a better gauge of where you stand.
Yeah. What is the species of varroa mite found in Trinidad?
Varroa destructor.
Okay, they are saying that they suspect it's the same species we have here too. Oh, the professor that came in, Dr. David Rubik.
Right. Yeah, well, I think varroa destructor is the one that's causing all the problems across the planet.
Yeah.
All right.
All right. Thanks for sharing. Thanks for your information.
Well, thanks for your input, Lyndon. Thanks for your input. Great
Yeah,
comments,
man.
great
Sorry,
input.
I'm late.
How is everything going in Ghana, man? I know you guys have had a lot of rain. Has the season finally picked up for you guys?
Richard,
The Long John is coming in, boy. Long John coming out. The Long John is. I need to find out from Gladstone. Our last Congress in 2018, some of the delegates from Trinidad took back some Long John seeds to plant. I want to know if they're germinated, if they're getting through with it.
All right, so gentlemen, let me just interject at this point in time. We still have some questions coming and it's getting rather late. It's already 9 o'clock. So let me ask David Small, if you could ask, my apologies to the two gentlemen. I know you all could catch up another time. David Small, can you please unmute and ask a question? David? Oh, it appears David does not want to ask any questions anymore. All right, so I'll open the floor up to any further questions. I know it's rather late and, you know, it's getting on. We all have to, you know, prepare for our next day. I would like to open the floor for any closing remarks from our presenters. Anybody would like to share or discuss anything? David, any last comments?
I would just like to say thank you so much. I really appreciate this discussion and hopefully a future working relationship. I think there's a lot of very intriguing scientific and beekeeping questions here that I'm happy to be part of.
Yes. Before we hand over to another presenter, Jorge, you had a question you wanted to ask? Jorge, you wanted to ask something? I see a hand up. I'm not sure if Jorge can hear me. All right, I'll hand over. Lena, any last comments, questions? Gladstone? Ryan?
No, I'm okay, thanks.
All right, thank you, Gladstone.
I have one thing to touch for. I think Harvey was talking about the introduction of stingless bees and pedial larceny and so forth. When you care for stingless bees, you don't really need much gears behind it. And I've known beekeepers that have stingless colonies right next to their house. So you shouldn't worry about somebody coming to steal your hives and things so forth when it comes to stingless bees. So that's basically about it.
Okay. Richard, anything you want to add?
Let's try and get Jorge back again. I think he's calling in from Grenada.
Okay,
yes.
Jorge.
Can you hear me?
Yes,
yes,
we can.
Okay, thank you. Finally, I solved my technical problem as usual. It has been several recommendations, several pieces of advice towards the topic of this very interesting meeting this afternoon, for which I am so grateful. And there is one point that I have seen for many decades, really, replicated throughout most of the countries in the tropical beekeeping practicing areas, that is common to most the small to medium beekeepers. And I am talking about the strength of the hives. People, wherever you go, think in terms of how many hives I have. They very seldom, if ever, think about, are my hives strong enough? The reason for my intervention in this topic is very simple. A strong hives for any purpose, production, protection, hygienic purposes, the physical effort needed for it, for any way you can look at it, are the only rational way to practice keeping anywhere. So Varroa control. Varroa is here already to stay, and we are not going to see a replication in our lifetimes of what happened to Apis cerana, where Varroa originated from, which they finally got adapted through millions of years of evolutionary process. we too short for that. Therefore, we have to deal with Varroa in a very practical, very sensible, very philosophical way. Gladstone mentioned something that to me makes a lot of sense. Once you see Varroa, you don't need to do anything. You see Varroa, you have to do something about Varroa, otherwise you are in trouble. I find that this drive to a so technical approach to the counting and the apistan and the other thing and the chemicals and things is a little more commercially driven, like so many other things in our third world countries that really defend vested interests of multinational companies and so forth. Okay, you know what I am talking about. It's necessary to be rational, to be down to earth. Think about a bee. Don't think about a beekeeper. Beekeepers in the world think in how many bottles of honey I can make, how much money I'm going to make, so how much equipment I need, and so forth. They seldom out from the viewpoint of what is the requirement of the colony, what about the timing of the year, what about my knowledge about the whole thing. If we start looking at it, like in many other fields of medicine and everything, in a more humane way, the profits in the medium to long term are going to be 1,000-fold. Besides, I appreciate a concept that Dr. Tarpy expressed in terms of the presence, and I know for a fact that the presence of Nosema in Trinidad and Tobago is something that is of long standing there. For decades, Nosema has been present in Trinidad. I don't know about the situation in Tobago, but Trinidad has been with Nosema for many years as a result of very unwise importations of bees from many countries, which is another topic that is to be discussed somewhere in another place. Therefore, if Nosema is present out there, why the commerce of honey and products from the hives is forbidden if they have the problem they want to prevent from inside most of the islands which don't have it? I don't see the rationale of it. I see, and I've seen it for half a century whilst I've been living in the Caribbean, that it's very easy to buy things from Trinidad, but very hard to sell things to Trinidad. So let's think about like that in a realistic, down-to-earth, cold-hearted manner. Let's forget about nationalism and things like that. We're talking about the region. Thank you very much. Goodbye.
Thank you.
Okay, all right, brother.
So I'm going to ask David very quickly. David, you have less than a minute to make your point or ask your question. I think David is having some technical issues. All right. And if there's no other information, oops. I just wanted to close with a quick summary of what some of the salient points that we had this evening. Basically, we've seen this evening that Varroa is a major vector for many viruses of bees, that including the acute bee paralysis virus and chronic bee paralysis virus and deformed wing virus, of course. We've heard that Varroa may be actually favoring more virulent strains of these diseases, and because of it, it's actually we're going to see more and more weakening of hives locally. And in order to be able to address this, we need to understand what we actually have in terms of diseases. So there's a need for greater monitoring. Of course, there's an important need for proper hive management, but most importantly, we need to be able to work with each other as beekeepers in order to eliminate the problem. And that means working together as a team in Trinidad and Tobago to address the prevalence of these diseases. The other thing that we saw is that Africanized bees can actually be negatively impacted by Varroa, despite popular belief. And that, we need to move away from the traditional thinking that Africanized bees will just
fight off varroa on their own and start looking at a more in-depth or more responsible way of dealing with the disease, not necessarily for the Africanized bee alone, but also for the interest of the other pollinators, which may soon be or may already be impacted by these diseases. There is little or no understanding of whether the list of diseases that we've seen in the bees is affecting the pollinator, other types of pollinators, other types of insect pollinators. And so there's a need for greater data collection and a better understanding of what plants are pollinated by which pollinators, so we understand the transfer of these diseases themselves. And we have actually used that opportunity to call on persons to participate in our first citizen science project for the night, our first recommendation for citizen science, where we want all of you all to become citizen science, which is to contribute to iNaturalist and the information that Lena is trying to collect in through the iNaturalist forum. We also learned that varroa is in all of the countries in the Caribbean. So in other words, potentially some of these diseases may actually be across the entire region, and some of the diseases are actually transported via honey. And as such, if there's a removal of the ban of importation on honey in Trinidad and Tobago, or restrictions or whatever is removed, that there's a potential movement of diseases if Trinidad begins to import honey. And being a center of economic, Richard had said industrial, but economic hub for the Caribbean, if the honey is imported to Trinidad and then distributed outwards, we realistically could become a source of potential diseases for the other countries as well. I really appreciate Jorge's contribution from Grenada, because he said something that's very, very poignant. Varroa is here and here to stay, and we have to learn to deal with them in a practical manner. And some of the ways in which we have approached or recommended treatment of varroa, as well as the fungal infection, the viruses that we spoke about tonight, was ensuring that you do proper and regular hive management, that you do regular varroa mite testing, and that you also treat your varroa mites in order to be able to ensure that you reduce at least the vector, therefore in reducing the spread of a lot of the diseases, including varroa. But there was also a recommendation for queen rearing and the development of more varroa selective hybrids of types of queens that would introduce greater resistance, genetic material that would allow for greater resistance, hopefully, to varroa into the colonies. A really excellent recommendation was trees for bees, but also ensuring that you have a variety of food sources, because we also do not know what chemicals are extracted from the from the plants, from the pollen. We do not know what compounds are removed in order to develop the immunity of the bees, and hence a wide variety is what will actually give you stronger hives. An interesting point that came out of this meeting as well is that the shape of pollen has an impact on immunity, a positive impact on bee immunity, and there's of course a need for greater work in pollen identification as well. And finally, where we cannot provide more trees of different varieties, maybe because of soil type or whatnot in your countries, you can look towards supplementary feeding as a potential way of addressing varroa and maybe even the viruses that they carry. So with that, I would like to say thank you very much. It's been a very, very informative session, an extremely long one. But what has been impressive for me is that we are still over 70 at this time of night. And I have to commend our participants for sticking it through with us. I know there's a lot of that was said, and there's a lot still to be done. So this is just the beginning. We have raised your interest, we piqued your interest. We have provided for you contacts for iNaturalist platform, where you can now start contributing wherever you see a pollinator on a plant. we would recommend that you definitely take that opportunity to share and become a citizen science through the iNaturalist platform. We've also shared with you how to test for varroamite. That video was shared. It's also available on YouTube off of the Iyanola Apiculture Collective's website. And that can give you some ideas as beekeepers on how you can test and ensure that you control your varroamite in your hives. But the last thing I've also done is that in the chat, I have put in a link to an Excel sheet where I have allowed persons who are interested in the second citizen science project for this evening, which is interested in doing a varroamite monitoring program for the entire Caribbean. I'm going to put back the link again. I've just shared it again. So I've just put up again the Excel sheet. So if you're interested in joining us in your country by monitoring our varroamite and you're willing to share the numbers that you've gotten on so that we could start plotting that information to build a case for why we need more funding to do the kind of work that we want to get done, that would greatly be appreciated. So feel free to put your name on the list and don't forget to put your contact information so that we are able to email you and keep you in the loop and in the process as we move forward with this project. So with that, I would like to thank my my co-presenters and panelists, and especially Dr. Tarpy. I'm so sorry, I did not think it would be going so late. It's 9:30 almost. Lena as well. Lena, thank you so much for your patience. Thank you, son, your poor baby, who has been waiting for you, I'm sure, whilst this whole process, this whole presentation is going on. Thank you to Gladstone, Ryan, who also has a young son, right? And also Richard, Matthias. And all of the participants for this team. This was an excellent opportunity, an excellent exchange. And we hope to publish, I believe, a small little booklet, you know, sort of giving a little bit of advice coming out of this. We hope to reach out to you beekeepers and our beekeeping fraternity across the Caribbean with the advice and the information that was shared this evening. So without any further ado, thank you very much and have a good night. Yeah.
Hands up.
Well done, well done.
Thank you. I'm seeing a lot of messages coming in.
Yeah, good show, guys. Good show.
Zena has left and has come back.
Well done, everybody.
Thank you, thank you. Yes, we'll email everybody a seminar evaluation form afterwards. It's too much to do on this hour. Really too much to do on this hour.
Well, we have to work on part 2 of this webinar. Yeah.
I agree, but I am all for part 2. I am all for part 1.1. Let's put a little, like just how I did the summary there. Let's put it all together in a little document or a little flyer, you know, let's put it out as a flyer or a joint flyer.
Have a pleasant night, everyone.
I'm for it.
Good night, Ian. We need to talk tomorrow when I see you.
Thanks, everyone. Great job.
Okay, all right. Have a good night, everyone.
Yeah, sure. Right, so.
All right, good night, all.
Good night. Good night, you all. Be safe. Godspeed. And yes, we will be sharing the recording. Unfortunately, it starts probably in the middle of Dr. Tarpy's presentation because I forgot. But yeah, we will definitely share whatever recording that we have. I will email everybody. Don't worry, I'll email you. And Vanessa, I'm really looking forward to assisting you all in doing some sort of varroamite testing in Dominica. I think it would be exceptional to get some data out of Dominica. Right? Good night, everyone.
Thank you.
Good night, all.
All the best.
Good night. Thanks very much.
Thanks for everything.
Not a problem.
Great show.